Welcome to the Microbiology and Immunology Unit-1 first video of clinical bacteriology. 1. Cell structure and morphological classification of bacteria. Cell structure of bacteria. Cell wall, made of peptidoglycan, gives shape and rigidity. Gram-positive bacteria have thick peptidoglycan layers, gram-negative have thin layers and an outer membrane. Cell membrane, semi-permeable phospholipid bilayer that controls substance movement. Cytoplasm, gel-like fluid containing enzymes, nutrients, and DNA. Nucleoid, region containing circular DNA, no membrane-bound nucleus. Ribosomes, 70s type, involved in protein synthesis. Plasmids, small, circular DNA for antibiotic resistance or other traits. Flagella, used for motility. Pili/fimbriae, used for attachment and conjugation. Capsule, protective outer layer, helps evade immune response. Morphological classification. 1. Cocci, spherical. Diplococci, e.g., Neisseria. Streptococci, chains, e.g., Streptococcus. Staphylococci, clusters, e.g., Staphylococcus. 2. Bacilli, rod-shaped. Straight, e.g., Escherichia coli. Curved, e.g., Vibrio cholerae. 3. Spirilla, spiral-shaped. E.g., Spirillum. 4. Spirochetes, flexible spirals. E.g., Treponema pallidum. 5. Pleomorphic. Varying shapes, e.g., Mycoplasma. 2. Bacterial growth requirements. Physical requirements. Temperature. Psychrophiles, 0-20 degrees C. Mesophiles, 20-45 degrees C, includes most human pathogens. Thermophiles, 45-80 degrees C. pH, most prefer pH 6.5-7.5. Oxygen. Obligate aerobes, require O. Obligate anaerobes, O is toxic. Facultative anaerobes, can grow with/without O. Microaerophiles, require low O. Aerotolerant anaerobes, do not use O but tolerate it. Moisture, essential for metabolic activity. Osmotic pressure, most prefer isotonic conditions. Nutritional requirements. Carbon source. For cellular components. Nitrogen source, for proteins and nucleic acids. Minerals, e.g., Mg superscript 2, Fe superscript 2. Growth factors, vitamins, amino acids, for fastidious organisms. 3. Principles of various staining techniques. Simple staining. Uses one dye, e.g., methylene blue, to highlight cell shape and size. Gram staining, differential. Classifies bacteria as gram-positive, purple, or gram-negative, pink. Crystal violet iodine alcohol safranine. Based on cell wall composition, peptidoglycan thickness. Acid-fast staining. For Mycobacterium, e.g., TB, with waxy cell walls. Uses carbylfuchsin, heat, acid alcohol decolorizer, methylene blue counterstain. Negative staining. Stains background, e.g., India ink, to visualize capsules. Endospore staining. Highlights spores using malachite green and safranine. For normal bacterial flora of human body. Skin. Staphylococcus epidermidis, Propionibacterium acnes. Nose and throat. Staphylococcus aureus, Streptococcus viridans. Mouth. Streptococcus mutans, Lactobacillus. Gastrointestinal tract. Escherichia coli, Bacteroides, Lactobacillus. Vagina. Lactobacillus, Candida albicans, fungus, Gardnerella. Importance of normal flora. Compete with pathogens. Produce vitamins, e.g., K, B1, 2. Stimulate immune system. 5. Bacterial culture techniques and culture media. Culture techniques. Streak plate, isolate pure colonies. Pore plate, quantify bacteria. Spread plate, even distribution on surface. Broth culture, for large volume growth. Culture media types. Basic media, nutrient agar/broth. Enriched media, blood agar, chocolate agar. Selective media, inhibits some, allows others, e.g., McConkey for gram-negative. Differential media, differentiates based on metabolic activity, e.g., lactose fermenters. Transport media, for sample preservation, e.g., Stewart's, Amy's. 6. Controlling microbial growth. Sterilization, kills all microbes including spores. Heat. Autoclaving, 121 degrees Celsius, 15 Ci, 15-20 mins. Dry heat, 160-180 degrees C for 2 HRS. Filtration, for heat-sensitive fluids. Radiation, UV for surfaces, gamma for equipment. Chemical sterilants, ethylene oxide gas. Disinfection, kills most pathogens, not spores. Alcohols, ethanol, isopropanol. Phenols, halogens, chlorine, iodine. Aldehydes, formalin. Quaternary ammonium compounds. Antisepsis, on living tissue. Antiseptics, e.g., iodine, chlorhexidine, alcohol. 1. Which of the following structures is responsible for bacterial motility? A. Capsule. B. Pili. C. Flagella. D. Ribosomes. Answer, C. 2 gram-positive bacteria stain purple because of A. Thin peptidoglycan layer. B. Presence of outer membrane. C. Thick peptidoglycan layer. D. Lack of cell wall. Answer, C. 3. Which of the following is an example of pleomorphic bacteria? A. Bacillus subtilis. B. Vibrio cholerae. C. Mycoplasma. D. Escherichia coli. Answer, C. 4. Mesophilic bacteria grow best at A. 0-20 degrees C. B. 20-45 degrees C. C. 45-80 degrees C. D. Above 80 degrees Celsius. Answer, B. 20-45 degrees C. 5. Which medium is selective for gram-negative bacteria? A. Blood agar. B. Chocolate agar. C. McConkey agar. D. Nutrient agar. Answer, C. 6. The primary stain used in gram staining is A. Safranine. B. Methylene blue. C. Crystal violet. D. Carbylfuchsin. Answer, C. 7. Acid-fast staining is used to identify A. Staphylococcus. B. Escherichia coli. C. Mycobacterium tuberculosis. D. Clostridium tetani. Answer, C. 8. Which bacteria are normal flora of the skin? A. Lactobacillus. B. Staphylococcus epidermidis. C. Neisseria. D. Bacteroides. Answer, B. 9. Which of the following is an obligate anaerobe? A. Staphylococcus aureus. B. Clostridium botulinum. C. Escherichia coli. D. Mycobacterium leprae. Answer, B. 10. Which bacterial structure helps in conjugation? A. Flagella. B. Ribosomes. C. Pili. D. Capsule. Answer, C. 11. What is the function of the bacterial capsule? A. Protein synthesis. B. Cell division. C. Immune evasion. D. DNA replication. Answer, C. 12. Which type of media supports the growth of fastidious organisms? A. Selective media. B. Basic media. C. Enriched media. D. Differential media. Answer, C. 13. The part of the human body where lactobacillus is commonly found. A. Mouth. B. Vagina. C. Skin. D. Lungs. Answer, B. 14. Which of the following techniques is used to isolate pure bacterial colonies? A. Pour plate. B. Spread plate. C. Streak plate. D. Broth culture. Answer, C. 15. Which method is most suitable to sterilize heat-sensitive fluids? A. Autoclaving. B. Dry heat. C. Filtration. D. Radiation. Answer, C. 16. Ethylene oxide is used for sterilization because it A. is a liquid disinfectant. B. is an antiseptic. C. kills only gram-positive bacteria. D. can sterilize heat-sensitive equipment. Answer, D. 17. Which disinfectant is effective against viruses and bacteria? A. normal saline. B. phenol. C. water. D. gelatin. Answer, B. 18. Which of the following bacteria has a spiral shape? A. E. coli. B. Staphylococcus aureus. C. Treponema pallidum. D. Klebsiella pneumoniae. Answer, C. 19. Which stain is used for visualizing bacterial endospores? A. methylene blue. B. safranine. C. malachite green. D. crystal violet. Answer, C. 20. What does an antiseptic do? A. sterilizes surgical tools. B. disinfects non-living surfaces. C. prevents infection on living tissues. D. kills spores in soil. Answer, C. Thanks for watching. For more videos subscribe the channel and hit the bell icon for latest update.